It's my great pleasure to be here. I'm going to talk today about a line of methodology that's really about fighting reference bias. And I'm going to talk a little bit about at the beginning about what reference bias is. And as you'll see, I know you recently heard from Dr. Benedict Payton, who's also probably talked to you about things like graph genomes and pan-genomes. And so I'm going to try to sort of take a different path and talk about a different and distinct line of work from that line of work, but I'll also do some comparing and contrasting with what's been going on in graph-based pangenome land. Okay, so let me start with the human genome, something that probably many people here have worked with, if not everybody. So at some point you've had to download it, or you've had to run it through a program that indexes it, or otherwise specified it to some bioinformatics tools. You've had to call it by one of its many names, like GRCH38 or HT19 or something like this. And the human genome, as we know, if we crack open these FASTA files and look at them, is a string. It's a collection of strings, right? Genomes are linear strings or circular strings. We've had this genome, in particular, the GRC reference genome we've had for many years. And we've had a pretty consistent complaint about the genome, which is that it doesn't represent the variability that we know exists among humans, right? So it's just one haplotype. It's just one haplotype assembly of human. And over time, we, of course, have learned more about the wider range of human genetic variation through, for example, the 1,000 Genomes Project, where this figure comes from, but also through many other projects, a few listed here. GBSBC, which is the Human Genome Structural Variant Consortium, the Human Pan-Genome Reference Consortium, which I'm sure Dr. Payton talked about, T2T, which was the consortium that did the recent gapless or mostly gapless assembly of the human genome. And so it seems a shame as time goes on and we have more resources like this, it just sort of seems like it's a pity that we're not using this somehow in our day-to-day reference genomes. That's one argument. I sort of call it the isn't it a pity argument, and that's one way of arguing for reference genomes. But I think the more positive and important way to argue is to point out the problems that arise when we use linear reference genomes, and that's what I would call reference bias. So I would use reference bias as a general term for all the sorts of things that go wrong in your bioinformatics analyses when you're trying to align reads to a reference genome and the reads might have non-reference alleles. So for example, here's a schematic of a reference sequence. I've got a few diamonds showing you where there's polymorphism in the population. And if you happen to have reads that match the reference genome, which is to say they have the reference alleles, then it's perfectly fine. It's a perfectly simple, usual problem for the read aligner to figure out that those reads go in those positions with respect to the reference. If the reads happen to come from a donor individual that has alt alleles at those three loci, then the read aligner has a harder time of figuring out that that's where the read belongs. Those alt alleles induce mismatches and gaps. It interferes with the aligner's heuristics, and it interferes with the aligner's ability to assign a good relative rank to different candidate places where the read might go. So you have instances where the read might not align at all, or it might align to the wrong place. Sometimes this phenomenon is confounded with what you would like to study. And so a classic example would be allele-specific expression. So let's say you're trying to study RNA sequencing data. Your ultimate goal is to try to figure out if you're getting more transcripts from the maternal or the paternal copy of a gene. And let's say the maternal and the paternal copy differ in terms of how dense they are with alt alleles. So maybe in this case, the maternal copy has 1, the paternal copy has 0. Maybe there's an ever so slight bias against being able to count and find those alignments to the maternal copy of the gene, but maybe it's just slight in this case because there seems to only be one alt allele interfering with our task. But what if I show you gene 2 and gene 2 is actually denser with alt alleles, and now maybe the aligner is completely confused. It can't find any good seed hits for its seed and extend heuristic. It fails to find some of the reads. My point is that when this does happen, when your ability to recover reads from one of the transcripts differs because of the abundance of alt alleles on that transcript, that's perfectly confounded with what you're trying to study, which is allele-specific expression. This, in fact, was the sort of place where the term reference bias, as far as I know, was coined. There's an early paper from 2009, early days of Solexa Illumina sequencing, where they pointed out that this was a severe problem in trying to study allele-specific expression, and they called it reference bias. However, it has other forms too. So for example, humans have regions that are so-called hypervariable regions. So you compare two humans, those regions the alleles that differ between two people tend to consist of more dramatic, longer, unshared sequences, maybe more structural variation. And therefore, if you use one person as the reference and the other person as your donor, and you go to align reads from one to the other, you end up with peaky, trophy sort of motifs in that area, where some places the reads happen to map well, and other places, which might be places where there was unshared sequence between the two, it doesn't align so well, and you get those troughs that you see in this picture. So it's a problem for hypervariable regions. Here's one more example I like to show. This is actually from the methylation world. This was looking at a few different mice of one inbred strain and a few different mice of another inbred strain, trying to compare their methylation levels. This is what's called a DMR, or differentially methylated region, where you can see the lines come apart in the middle. However, it's spurious. It's not real. And it's caused purely by genetic differences between the mice, not epigenetic differences. And so you can see that not only do you get the spurious result, but as an additional nightmare, the direction of the result switches depending on which of the two inbred strains you use as the reference. So I almost don't like to show this slide because I feel like good bioinformaticians will have nightmares tonight thinking of this example of spurious signal that switches direction depending on what reference genome you use. But these are some examples of where reference bias affects what we can do day to day. So we should attack reference bias because we want to avoid living in a world where the sequencing-based diagnostics and therapeutics that we develop are differentially effective depending on whether the person we're studying happens to have alleles that match the reference genome or not in crucial places. And this is not just something bioinformaticians worry about, although it has been a longstanding problem that we have worried about. It's even cropped up in other places in the press, for example, this STAT article, which is very specifically about the reference bias problem. Okay, and by the way, the T to T assembly in no way solves this problem, because after all, the T to T assembly is actually 100% European. So it is in no way more representative of world populations than, for example, the GRC genome that we've been using for longer. Okay, so this is, I think, the real motivation for why we need to attack reference bias. So what am I going to talk about today? I'm going to first try to distinguish the kinds of topics I'm talking about from the graph pangenome set of work. I'm going to return to it later, but I think I first want to push off and go off in a different direction. And I want to motivate why we've gone in a different direction than indexing graph pangenomes. Then I'm going to talk about some details about why what are the particular mathematical approaches underlying our ability to build good linear pangenomes. I'm going to talk about the Burrows-Wheeler transform, which was brought up in the introduction. Hopefully you're not yet tired of hearing about the Burrows-Wheeler transform, because I'm going to have more to say about it today. And then I'll talk about how the idea of R-index emerged, what R-index can do today, and then how we're going to hopefully be able to continue to push R-index and the Burrows-Wheeler transform further into the future. And I'm going to circle back to graphs at the end. So let me start with why we were compelled to look at pangenomes that are linear. Linear in the sense of they consist of collections of linear reference genomes. They are not collapsed into a graph. They are separate. They are left alone. Okay, well, here's a picture of a variation graph. This is from the VG paper. And my point here is that we're all trying to solve the same problem, right? So like the VG graph, the pangenome graph that comes from the VG project, is an example of an index data structure that is trying to overcome the reference bias problem. This is the stated purpose for the graph pangenome. Early in the days of graph pangenomes, some students from my group investigated a couple of questions about if you have a graph pangenome and you're going to add variation to it, what variation and how much variation should you add? Should you add all of it? Or should you perhaps be picky and only select some of it for inclusion in the pangenome graph? So let me start with the paper on the left. This was the paper describing the so-called Forge tool for prioritizing variance. This is a relatively complicated graph. I'm going to actually distill it down in a moment. Actually, I'll just go ahead and do that now. I really just want you to concentrate on one line and one dot in this picture. What that line represents is, as we go from left to right in this picture, we're building pangenomes that are more and more inclusive of variation that was in the 1,000 Genomes project. So we started at linear. Leftmost point is a linear genome. Rightmost point is include absolutely everything from the phase 3 call set from the 1,000 Genomes project. Every point in between is that we're selective. We just start with the most frequent alleles, the highest allele frequency alleles, and include more and more and more as a percentage of all the alleles in the BCF file. Okay, and so what we find is this very clear peak, then come back down, right? And we reach the peak relatively quickly. So the peak in terms of, I should have explained the y-axis, the peak is representing basically what fraction of the reads were aligned correctly when you use the corresponding pan-genome. So this is like a way of measuring how good is the pan-genome at removing the penalties we wanted to remove for alignments that come from known alleles. So the peak accuracy came at something like 10% of the variance from the BCF, which if you look at that as an allele frequency cutoff, was around 5%. So it just happened to include what we might call the common alleles from the 1000 Genomes Project call set. We reached that point, I would say, relatively quickly. And then once we reached that point, something started to go wrong such that our ability to place reads correctly actually deteriorated after that point. And I think that this sort of makes sense when you think about the two things that are in competition here. So on the one hand, adding more variation to the graph pan-genome takes away the penalties associated with those alt alleles that we saw before. And this is good because it takes away the bias. But then once you've added the alt alleles that are in common with the individual, or substantially most of them that are in common with the individual you're trying to study, adding more doesn't help a lot. In fact, it could help you, it could hurt you by making the pan-genome representation more and more ambiguous, because the more variance you add, the more repetitive the overall genome gets. So there's a point where the con outweighs the pro, and that's when this line starts to go back down. The other thing I want to show you is that we made as a reference point, as a kind of benchmark, a personalized reference, which is to say we made a reference that had exactly the alleles that were from the individual that these reads were simulated from. They were, I believe, from NA12878. And so that is a personalized genome. It has exactly the alleles that are in the donor and no more than that. And that achieves somewhat better on this measure than any of the graph pan-genomes that we tried. Kind of no surprise here, right? If your graph contains exactly the alleles that are in the individual and no more, then you're getting all pro and no con. And so you're doing a little bit better than any of the graph pan-genomes. Okay, so the pan-genome accuracy approaches, but doesn't quite reach that of the personalized reference genome. So this overall, I would say this surprised us. We thought the graph pan-genome did quite well, but we were surprised at how sensitive it was to the particular set of variants that you include in the graph pan-genome. And we thought that was a pretty small proportion that ended up maximizing the measure on the vertical axis. So we kind of asked ourselves, well, what if we go to the other extreme and we start with a single linear reference genome and add a few more linear reference genomes? So not all the way to a graph, you know, chock full of 1000 Genomes Project alleles, but just a handful of linear reference genomes. And so this is a result from another paper describing a method called reference flow. I'm going to draw a line here corresponding to where we would be if ref and alt alleles, there seemed to be no bias either toward or against reference alleles. And anything above the line is somewhat biased toward reference alleles. That's reference bias. So the degree to which we're above the line is a measurement of reference bias. And these are these sets of boxes. There's 4 clusters of box plots here, and they come from, here's where the font issue is going to get fun. They come from 4 different kinds of pan-genomes. The linear genome on the left, which does poorly. No surprise. A set of 6 linear reference genomes, one that we aligned to first that was basically a major allele reference, and then 5 more that were derived from each of the 5 superpopulations from the 1000 Genomes Project. That's the collection of 6. Then a pan-genome graph that was VG. So that was the VG graph pan-genome came next. And then again, personalized. As always, personalized does best, right? So that had exactly the alleles from the donor individual. But surprisingly, even just a handful, 6, I'm going to call that a handful of linear reference genomes actually competed very favorably with the much more complicated and at the time slower and using more memory graph pan-genome approach based on VG. So I thought that was remarkable. However, it was not necessarily a good idea to go ahead and use these 6 reference genomes in your analyses, in particular because that's only a handful and you're going to miss even, it says miss rare alleles, but you're even going to miss common alleles when you only have 6 reference genomes in your so-called pan-genome. So it's far from being comprehensive. So the question became, how big can we go? Can we stuff many, many, many linear reference genomes in a compressed index and compete, but without ever making a graph, right? That was sort of, and furthermore, we might not have to go much further than 6, but we would like to be able to go, maybe to 100 or maybe to 1,000 or maybe to 1,000,000 eventually. Okay, so now I'm going to move on to how maybe we would be able to achieve this. Index many linear reference genomes using some tools that are well known to the community by now because we've been using them for more than a decade, like the Burroughs-Wheeler transform and related things like the FM index, and something newer called the R index, which I'll get to. Okay, so here's the part where I get to show you the Burroughs-Wheeler transform slides. Love the Burroughs-Wheeler transform, loved it for a long time, so I always enjoy showing these slides. In a sense, the Burroughs-Wheeler transform of a string, so like the string T on this slide, think of it as maybe a genome sequence. Also, we do this funny thing where we slap a $sign on the end of it. Don't worry too much about that. It's a mathematical convenience. It's not there to confuse or annoy you. The Burroughs-Wheeler transform is really just a way of taking the letters of the genome and putting them in a new order, a different order. In some sense, the best order that you could possibly put them in, which sounds strange because after all, T is the order that evolution put them in. Why are we trying to change that? But the order on the right is the best order if your goal later on is to look for patterns inside T. It's the best possible order you could pick if that's your goal. So the Burroughs-Wheeler transform takes the letters of T, and in particular, it reorders them according to the alphabetical order of what comes to the right, just to the right, inside the string T. Just as another sort of intuitive motivation, why do we care about taking T and putting it in a different order? Well, I think that's pretty intuitive. We do things like this a lot. For example, if I gave you a deck of cards and said, let's play poker, you would shuffle them, right? Because that's the order, random order is the order that's useful if you want to play a game. If I gave you a deck of cards, I said, there's 51 cards here, please tell me which one's missing. You would put them in another order. You would probably put them in rank and suit order so that you can then scan and very quickly figure out which card is missing. This is the same sort of idea of why we want to put a genome in a different order, because it's more useful if our goal is to look for patterns in the genome. And, we're bioinformaticians, so that's totally our goal. We do a lot of looking for patterns inside genomes. Okay, so this is actually another maybe more common way of representing what's going on with the Burroughs-Wheeler transform. It's as though we took our text, we took every different way of rotating the text, sort of like chopping it into two pieces and switching the two pieces. And then we took what's called the final column of what's called the Burroughs-Wheeler matrix. The particulars are not that important. The main thing that I want to convey is that once we have this Burroughs-Wheeler transform, we have something that we can use as an index. It's a little tricky to see how it can be used as an index, and gory details are available on my YouTube channel. But I'll just show you, at least pictorially, what it looks like when we use it as an index. We're going to, let's say this is our index here, mostly consisting of just the two columns that I've highlighted here. And when we go to match a string against this index, we're doing an iterative process where we start with our string. Let's say our pattern P is that ABA that you see on the screen right now, the little weird thing next to it. And we're going to, in a series of steps, find the rows that begin with longer and longer suffixes of our pattern. So here's the pattern of length 1. Then in the next step, we find the rows that begin with the suffix of length 2, which is BA. So there are those two rows at the bottom that begin with BA. And in the final step, we find the rows that begin with the full pattern ABA. And then usually we do one more thing, which is we try to figure out, well, where were those? In other words, what were the locations of those two ABAs with respect to the original sequence? And if I just gray out most of the screen, I just want you to concentrate on the fact that we're doing two kinds of things here, two different kinds of computation. The stuff we do at each step to match longer and longer suffixes, I'll call those match queries. And then the stuff we do in order to figure out where they occurred with respect to the genome, and I'll call those locate queries. Okay, so this was the very quick version, but the main thing I wanted you to distill is we're using this as an index. We're able to find patterns inside the genome. This is the basis for tools like Bowtie and BWA and tools you probably used, and also increasingly used for things like genotype imputation. It's also used in the VG aligner that probably Benedict spoke about when he was here. So these same underlying queries power a lot of different things. This is one of the two things that the BWT does really well. It creates indexes. Related to that, the other thing that the BWT does well is it compresses. Okay, so what do I mean by that? Well, the BWT, in the process of putting the string in this new order, where all the letters are ordered by their right context, what it tends to do is it tends to take underlying repetitive structure that's in the original string and bring it sort of to the surface, right? It might be there, but spread from here to here to here, and it's not immediately easy to see. And what the BWT tends to do is bring it to the surface so that it's immediately easy to see. So for example, here's a string I have on this slide. It's got a few different versions of the word rectangle or similar. So if you think about it, the three R's that start those three words that are basically rectangle have the same right context, right? They all have rectangle or something like it just to the right. So if I put the letters in a new order according to the alphabetical order of their right contexts, those three R's are going to come together in the Burroughs-Wheeler transform string. Okay, so there they are, and because of the font issues, the arrow is pointing in the wrong place. But anyway, the arrow should be pointing at the three R's. So it tends to bring characters with similar context together. And so when there is repetitive material in the original string, you get what are called runs, right? We brought the same character together in a run of three R's. Okay, let me show a more dramatic example that's even more repetitive. So here's my repetitive string, and I'm going to apply the Burroughs-Wheeler transform to that, and what I get is what you see just below that. As you can see, like big, long stretches of the same letter over and over again, because again, we've taken that internal repetitiveness and brought it to the surface. If I were to take the BWT and try to represent it in as little space as possible, a clever thing to do would be to run length encode it, where instead of writing www 9 times, I just say W, don't have my cursor, I see W and I say comma 9, right? In other words, W, but repeat it 9 times. And that would be so-called run length encoding. And that would be a wonderful way of compressing this string. This string isn't a pan-genome. Maybe we're getting closer to it looking a bit like a pan-genome, since there seem to be different genomes in it, right? Sort of like 3 things in it. Now, let me just add a little bit of genetic variation, because that's what we want to do when we index pan-genomes. Just take a quick note of these statistics. We've had 15 runs, and the average run length was 4.3. So now here's another example where I added some wacky typos, sort of in imitation of genetic variation, as though these were 3 genomes in a pan-genome. I'm going to do the same thing. I'm going to find its BWT. And I end up with a similarly kind of runny, several long runs version of the string after applying the BWT, though it actually didn't compress quite as well this time, which might not be surprising because there's more unpredictability to the strings now that I've added these mutations to them. And so now the number of runs in the Burroughs-Wheeler transform is 21, and the average run length is 3. Still respectable. There's 3 sequences here, so a compression ratio in a way of 3 is pretty good. But it went down. It used to be 4.3, went down to 3. Okay, so the question that we might have is, instead of silly examples like this, what about human genomes? Like, I want to make the analogy. What happens when we take many human genomes and put them into the Burroughs-Wheeler transform? It's going to compress them, but how much? Is it really going to be effective at compressing human genome sequences? Okay, so here's some data that's from a paper where we measured this. So we took increasing numbers of individuals from the 1,000 genomes phase 2 call set. These are actually diploids that we're adding. So I have that column that's labeled number, but those are sort of pairs of haplotypes that we're adding in each row. And so N, the column labeled N, represents the total length of all the sequences that we're indexing. And then the column R is how many runs are in the Burroughs-Wheeler transform after we compute the Burroughs-Wheeler transform. What you'll notice is those columns are behaving very differently, right? The N column is, as you would expect, right, by definition, increasing linearly. If we add more genomes, the total length of the pan-genome is getting longer linearly. R is growing incredibly sublinearly, right? That's very, very sublinear. It's growing much more slowly, right? Even that first diploid, right, it only results of an R of about 3.2 billion, presumably because the 2 haplotypes are similar to each other. So we ended up with a number of runs that was roughly half the total length of that diploid genome. So this is what gives us heart, that if we can apply the Burroughs-Wheeler transform first, and if we can find a way to live in R space somehow, an amount of space that's proportional to R, not proportional to n, that would not be good, right? If the amount of space our data structure was using was proportional to n, we would fall off the table pretty quickly because human genomes are big. But if the amount of space we're using is proportional to r, if it's big O of r space, then we have hope of getting to being able to index many human genomes. Okay, well, the theory community obliged us because they've been working on this problem for quite some time, the computer science theory community. So the paper that furnished the kind of ideas that went into Bowtie and BWA, that FM index paper, appeared in 2000 in a computer science conference, the Foundations of Computer Science conference. It did not achieve big O of R anything, right? Those runs were there in the BWT that it would compute, but it wasn't yet thinking in terms of trying to exploit those runs in any way, except for some mild compression. And it didn't really achieve anything better than big O of n in terms of how much space was required to execute those two queries that I mentioned on the previous slide, right? I said those were the two fundamental queries. We couldn't really do either of those in big O of R space. A few years later, in 2005, it's cut off, but it says 2005 there was a new set of work that figured out how to do one of the two queries in big O of R space. But the other one was still in big O of n space. So still, it wasn't incredibly useful as a data structure for genomics. We were still waiting for it to be able to handle that other query. And then many years later, 13 years after that, there finally was a team, their names are in the bottom citation here, that managed to get both match and locate queries to be serviceable in big O of R space. And this was published in another big computer science conference called SODA, just in 2018. So relatively, not long ago, relatively recently. And so it was at that point that the dream of being able to compress and index and query linear pangenomes really started to feel like, oh yeah, we can do it, right? The theory's there, we can do it now. And in fact, the first author of that study, Travis Gagey, immediately started emailing people like me and others and to be like, hey, guys, I know there hasn't been a lot lately in the theory world that you've been excited about, but you really should be looking into this. We think this result means you can do more than you've been doing with pangenomes. So we started to work together. And there's Travis in the middle there. That says Travis Gagey. And we started working together. They were already working on another very important problem that I'm not going to emphasize today, which was the construction of the compressed index. They had a clever idea that they called prefix-free parsing, which is kind of still the standard for being able to build large pangenome indexes out of linear references. And as a team, these folks and I, Tahir having been in my group, we worked together to apply it to genomic sequences to see, well, if we have this wonderful big O of R data structure, can we show how it competes with existing tools that are not big O of R? And so we did some comparisons. This is not my favorite slide to show because it's very busy, but the main thing to look at is toward the right-hand side of this plot. You see that dark green line that's going up, and then it disappears. That's Bowtie. So that's the size of a Bowtie index built over more and more copies of human chromosome 19, copies, which is to say different individuals' versions of chromosome 19 from the 1,000 Genomes Project. It eventually, as expected, because it's big O of n, runs out of memory and it doesn't run anymore. Yeah, question?
You talk about space complexity. Is time complexity not of interest, or is it just all the same patterns?
It is roughly, for all the queries on my previous slide, it is roughly constant. But there's actually some subtle trades where, like, for example, with the R index, it's not exactly big O of R space and constant time. There's actually, you can trade one way or the other, but you can't have both. But roughly speaking, which is to say, ignoring some mild log factors, all of these queries can be done in constant time. So everywhere where I had a label on the previous slide, just imagine we did that in constant time. And I'll actually, there's another plot next, which shows a time comparison of some of these same tools. So, okay, so Bowtie falls off the table eventually as expected because it's growing linearly. These other lines, and you have to, these are logarithmic scales here, so it's always a little bit tricky to interpret, but the fact that those sort of brownish looking circles ended up below all the triangles is a sign that our index was able to achieve better compression than a rival compressed index based on Lempel-Ziv compression, right? So these are just two different strategies for compression pitted against each other. Eventually, the R index strategy won. We also did a comparison, as I was just saying, based on timing, where we asked Bowtie and R-index to do combined match and locate queries for a bunch of patterns. And then we timed how long it took. Bowtie, which is no surprise, it's an attuned piece of software. It's been used for years and years, is very fast when the number of sequences in the collection is small. But there is eventually a point where it, again, first of all, fails to even build the index. That's where it falls off the plot, but then also becomes slower than the R-index for its locate queries. So it was competitive, right? So what we found was there's potential here, right? It's not winning in every category and at every size pangenome, but there's a lot of potential here for it to be a leading tool. So that's sort of the story of how this alternative way of doing things emerged. And just for re-emphasis, there's no graphs here. Right? The strategy really is to take a bunch of genomes, maybe they are all the genomes from the Human Pan-Genome Reference Consortium, for example, concatenate them, and then index them, right? And so there's no building of a graph at any point. Okay, so with this approach, what can we do today, right? That was sort of 2019-ish that we had those demonstrations that I just showed you. What have we really been able to do with it? Well, I'm going to tell a couple stories now that have to do with specifically classification, because we were excited in particular for applying these methods in areas where we knew we needed to be able to classify quickly some incoming sequence against what was potentially a pretty large pangenome, so like a lot of related sequences. And so we thought of adaptive sampling for nanopore sequencing, because maybe what you're trying to do is you're trying to use the nanopore to sequence just the reads of interest, but you also have a lot of reads that are not of interest. And so what you need to do is very quickly do some sort of pattern matching to figure out, should I eject the read or should I continue to sequence this read? So we tried to apply it in this problem setting. And so there was some work that was done by Max Rossi, who I saw was in the Zoom. So I have to take out all the critical things I was going to say about Max in this presentation because he's here. So Max did some very early work on being able to adapt the R-index to do a somewhat more sophisticated query than the one that I showed before, which is to say a matching statistics query. I'm afraid the alignment is all off for these strings, so it's a little hard to interpret what I have on the right there. But the idea is that you are, in essence, going along the pattern base by base and computing the longest matching half mem in one direction as you go. Okay, so you're extending the match as far as it'll go in one direction. This is a useful vector of statistics if you want to know about how two sequences relate to each other. The large numbers correspond to places where they seem to match well, and the small numbers are probably noise and where they don't match well. And also from the matching statistics, you could derive what are the MEMS, the maximal exact matches. Roughly speaking, it corresponds to the local maxima in the matching statistics vector. So Max found a way to adapt R-index to produce these vectors. And so these vectors are, in some sense, if we can compute them in kind of like an online fashion, like base by base, let's get the next number in this vector, it allows us to quickly come to a conclusion about whether a read does seem to or does not seem to match the database. I'm just want to make sure nobody's saying they can't hear me. Okay, good. Everyone's good. So we adapted that approach, which was called MONI, developed by Max, and we developed a new approach, which we called SPU-MONI, which was streaming pseudo MONI. And we took one slight tweak to the idea of computing the matching statistics. We said that's a little bit too much effort. We instead are going to be satisfied with computing something else called pseudo matching lengths, which what they tend to do is if you have a variety of different lengths of matching statistics, they kind of, in some sense, reduce your dynamic range so that the short matching statistics kind of get rounded off close to zero, and then the longer ones still sort of peak up. And we were able, why did we do this shift? We did this shift because this allowed us to compute the statistics in a more online way, base by base, as the data was coming off the sequencer. Okay, so when we applied this in a setting where other tools had been applied, a setting that looks like this. So we've got sort of data that's coming from a nanopore. We are taking the bases as they are coming off the nanopore sequencer. They have to be base called, right? So we're taking the base called bases from the nanopore sequencer. We get them in batches. And as the batches arrive, we are running our algorithm to compute the next several pseudo matching lengths. And then we have to decide, we have to make a classification decision about whether to eject the read or not. And the way we end up making that decision is based on comparing distributions of pseudo matching lengths. So you can imagine that if the read is present in the database, which is to say the database has sequences that are either similar or identical to the DNA from which the read originated, I'm going to get some nice long pseudo-matching lengths. Maybe not wonderfully long because of sequencing errors, but they'll be longer. Whereas if I compared that to the kinds of pseudo-matching lengths I would get if I was using a database that had completely random sequences not related to my read, those would be shorter. And so in some sense, we're asking, is the distribution of pseudo-matching lengths shifted with respect to what we would expect in a null setup, where the database is unrelated to the read? Or another trick is you can just take the read and flip it around, put it in the reverse direction as your kind of null set of pseudo-matching lengths. Okay, so if we are finding that there is a shift between the two, that tells us that the read seems to be present in the database, which influences our decision about whether to eject the read or not. And when we compared this approach to some existing approaches, like for example, the Minimap2 based approach that was proposed in the ReadFish tool, popular tool for doing this exact adaptive sampling application, we had comparable accuracy. We had comparable accuracy, and we were able to do it in a much lower memory footprint and with a smaller index and faster. And that's when we were using these tools with a database that was a pan-genome, right? In other words, we were pan-genome, which is to say many, many strains of the same species. So we were doing a mock community experiment here. So there were various bacteria and yeast, and we had many, many strains of those bacterial species and many, many strains of yeast in the database here. Okay, so comparing to a tool like Minimap that's doing a lot more intense alignment work, it turned out that the matching statistics that we could compute in a fast streaming fashion with Spumoni were in some sense just as able to do the discrimination between what should be ejected and what should not be ejected. So this was, I would say, this is only binary classification, and it's a particularly useful, perhaps even only useful in situations where the database has many related sequences, not just a single sequence. But it was an early success. So with my remaining time, I just want to talk about a few ways that we have since then pushed it a little bit further in a few directions. And then I also want to talk about future directions. And I've organized this part as a series of kind of like, let's think about a spectrum from this to that. So I've got 3 different kinds of let's think about a spectrum from this to that to sort of frame what the current and future work is all about. And who doesn't like Pink Floyd? So, all right. So number 1 is I showed this picture before. This was the result from the Forge experiment, where we found that the linear genome didn't do so well. That's like the green dot all the way at the left. The ideal pan-genome, the one that had exactly the right amount, the sort of Goldilocks amount of variation, did much better. But the best was the personalized. And this repeats itself in study after study. If you can build a personalized reference genome, you in some sense should, because that will avoid reference bias with the ideal least amount of added drag of having too many genetic variants in your representation. So in some sense, we know what the best option will be in many cases. It will be a personalized reference genome. Now, it's not always practical to build a personalized reference genome. It, first of all, takes effort, right? Computational effort that you're not used to spending, right? We're used to sort of picking up a reference genome from the shelf. Maybe we've already built an index over it, so we don't even have to build the index. It's already done. But this way of thinking would be, well, if I have a new, let's say, whole genome DNA sequencing data set from an individual, and I want to do a super good job of analyzing it with no reference bias or minimal reference bias, what I should do is sort of look at some of the reads, maybe slurp down some of the reads, not necessarily all of them, try to figure out, okay, what roughly speaking are the genetic variants this person has? You can do this with genotyping together with genotype imputation, because we have wonderfully fast genotype imputation tools now and pretty large reference panels. So if you just take a subset of the reads, let's say 1% of the reads, put it through a genotyper, you'll get rough genotypes here and there, but you put it into imputation software, you usually get a pretty solid, good, personalized, imputed diploid reference genome. If you then go and take all the sequencing reads and align to the imputed personalized reference genome, that is close to the personalized scenario from the previous experiments. Not exactly the same, because you're not going to do a perfect job of genotyping or imputing, but you're also not going to waste time representing the huge number of alleles that are in the 1,000 Genomes Project, but not in your individual. And indeed, with a strategy like this, where we used a genotyping tool that we developed called Rowboat, together with an existing imputation tool called Glimpse, which is quite excellent, And then we aligned all the reads to a personalized reference that we made using giraffe. So this was actually a giraffe graph, but where the only forks in the road in our graph genome were the heterozygous variants for this individual. We had better downstream variant calling accuracy. You could do measurements up or downstream. You could do an upstream measurement of reference bias or a downstream measurement of variant calling accuracy. We always did better than graph pangenomes built from a generic, like for example, 1,000 genomes pangenome, and definitely better than linear aligners too. So for all the pangenome talk today, I think it's important to remember the ultimate tool in avoiding reference bias is a personalized reference. Okay, so back to the pangenome space, because sometimes we can't build a personalized reference, so we do need pangenomes. Starting from the R-index Spumoni idea, we've gone in two different directions in terms of how to make it faster, because although we were much faster than the Minimap2 based approach, there's a lot of other tools out there that are K-mer based. So I'm going to name a few: Kraken, Fulgor, Salmon, Callisto. Those are blazing fast, and we were not competitive with those tools. So we needed to move further in the direction of more speed, please. So we took two different approaches for this. One of them was lossy, which was to use minimizer digestion, and that yielded this tool called Spumoni2, which I'll talk about first. Then we had another approach, which was lossless, but did a radical reorganization of the data structure in order to achieve fewer cache misses. So in Spumoni2, the idea is quite simple. I'll go through it quickly. We simply minimizer digested both the reference and the read before engaging in the pattern matching problem. This did two things. It made the reference smaller, but it also reduced the number of iterations of our inner loop because it made the query smaller. Further, once you minimize or digest, you have the option of shifting your alphabet to be the minimizer alphabet instead of the original alphabet, which we also did, which provided an additional boost in speed. That's the very short version. And that made a tool that we called Spumoni2 that was even faster with comparable accuracy than Spumoni. That was one direction we went in. The other direction I think is also extremely interesting, which was there was a later paper describing not the R-index, but a competing kind of data structure called the move structure. Came from two researchers in Japan named Nishimoto and Tabe. And they proposed it as a way of achieving a better combination of theoretical bounds compared to what the R-index was able to achieve. But they did something else too, which is that they radically reorganized the structure to have excellent, what computer scientists like to call locality of reference, which is to say it was very predictable where you were going to touch the data structure in every iteration, not across iterations, but within iterations, it was very predictable. And that meant that compared to competing data structures, the number of times you would have to wait for main memory to deliver the next bit of memory that you wanted to access was greatly reduced. In other words, we had fewer cache misses. And that in turn led to much faster speed, more than an order of magnitude speed improvement compared to Spumoni. So that's a preprint available now. So you can check out our work on the move structure. Okay, with my final minutes, I'm going to go to the third and final of my spectra that I want to talk about, which is the string versus graph spectrum. I love graphs, and we do a lot of work on graph pangenomes too. So I'm not a partisan or anything. But I do want to talk about why we shouldn't necessarily be comfortable with either extreme of this graph or string spectrum that I talked about today, why we might want to live in the middle instead, and what are some of the options for living in the middle. So I think that the paper that really helped us to think about graph pangenomes the most was this one, another breakthrough by these folks, including Travis, who I mentioned a few times, because they finally defined this thing that they called Wheeler graphs, also sometimes called Wheeler automata, which are not just string, in other words, the BWT has been applied to strings, but this applies the BWT to a larger class of mathematical objects that include many trees and some graphs. And for a long time, we didn't quite know what exactly was the class of graphs to which these methods could be applied. It was clear they could be applied to some and not to others, but we didn't quite have the dividing line between these two types of graphs. They proposed the definition that defined the dividing line exactly. Things that have these properties are so-called Wheeler graphs. Things that don't have these properties are not. The Wheeler graphs are in some sense the ones that are amenable to indexing, and the ones that are not Wheeler graphs, less so, right? We have some kind of blow up here or there if we attempt to index them. And so The graphs that are practical that we use day to day, including the graphs that are behind VG, which is a combination of de Bruijn graphs and other Wheeler graphs. De Bruijn graphs happen to be Wheeler graphs. There's other Wheeler graphs as well. That's an application of this idea. And it's an application where first you build a kind of, I would say, multiple alignment of all your genomes, and then you do a bunch of collapsing to turn it into a graph. And you end up with sometimes a Wheeler graph, sometimes not. And it depends on how you do it. And there's a bunch of tricks for trying to get it to sort of cram it into a Wheeler graph form. So what I want to say about this, I had this slide, which at the time seemed like a good idea. And then I rehearsed it this morning. And I felt like Charlie Day from It's Always Sunny in Philadelphia. So I'm going to summarize it in just a few words, which is that if we do things the fully graph way, there's actually a lot of drawbacks that we can encounter that come from the fact that when you do multiple alignment, you're usually giving something up. You usually have to give up, for example, on a tricky class of structural variants that interfere with your ability to build that multiple alignment. Or you give up on the multiple alignment and you build a de Bruijn graph instead, and that gives up on the sort of coherence of the coordinate system, because the de Bruijn graph likes to glue things regardless of where they are in the coordinate system, and you get kind of cross-genome gluing, which is not so good. So anyway, there's a combination of reasons why starting with a sort of biological notion of what the graph should be runs you into a few problems. Maybe they're all solvable, but they have not been solved to date. But the string angle, the one I've been talking about today, has its own problem, which is that it doesn't do any of the kind of collapsing, except in the size of the data structure, right? The data structure is big O of R, so that's great. But it doesn't do any of the conceptual collapsing that the graph genome does. It doesn't collapse the coordinate system for you. And that actually leads to a lot of problems downstream, more locate queries, all kinds of other issues like that. So I think, in my mind, I think the ideal point is not going to be at either extreme. It's going to be something closer to the middle. And if I had to propose one idea today about where we could go next, it would be to use this very beautiful idea called BWT tunneling. And the idea here is you start with a string. A string is a graph, right? It's just a really simple graph. So you just start with a string, which is a graph. And then you do some strategic collapsing, which is to say you collapse some parts of the string that have identical labels. And you can even do this hierarchically so that you get different levels of collapsing. It turns out every string is a Wheeler graph, and every point of collapsing at any point during this process, any tunnel collapse, results in another Wheeler graph. So basically, you're never escaping Wheeler graph space at any point in this process, but you are getting the same, in some sense, the same collapsing action that the multiple alignment of the VG graph got you. But it's all mathematical, right? There's no biological intuition besides that there's homology between the sequences. There's no biological reasoning behind what we're doing. We're just mathematically picking the best ways to collapse the graph. And I think that could result in a graph that's Wheeler for sure. That's guaranteed. That's just guaranteed according to the properties of the graph at each point. And it's collapsed, right? And so it's a little bit easier to deal with in terms of providing a coordinate system. Not perfect, but it's better than what we currently have with R-index. But we don't run into those problems that you have with multiple alignment, where inevitably you have to do some kind of violence to the sequences to get them to fit into that multiple alignment together. So, conclusions. First of all, I think that when used with multiple references, it turns out that linear aligners that are based on our favorite sort of bow tie BWA type principles, they actually do a pretty good job of avoiding reference bias. And they're competitive with the larger graph-based representations, which is interesting, right? It means that the linear regime didn't fail us quite as drastically as we thought it did. It turns out we just needed more sequences. More assemblies are coming, right? The Human Pan-Genome Resource Consortium is going to give us so much more than they've given us already. They're going to give us tons and tons and tons of human genome assemblies to very high quality. They will not necessarily enjoy being put in a multiple alignment together. They might be completely refractory to living in a multiple alignment together. And that is why I think we need additional methods like the ones that I talked about here that let genomes be linear, right? There's no such thing in nature as a graph genome, right? All genomes are either a line or a circle. And so these are methods that sort of let them be themselves, do not remove any structural variation at any point, keep it all in the representation. Okay, all right. And then the last thing I wanted to say is that I really love this field for the way that the theory and practice have been growing together. So the computer science community has injected all these wonderful ideas along the way and sort of made a lot of these things possible, including what the VG project has been able to do. If you are disappointed that I didn't talk in as much gory detail about BWT or any other ideas, I have a lot of videos on my YouTube channel, including an indexing playlist where I have an amount of material that can only be described as too much for you to enjoy. So with that, I want to thank everyone who's worked on this work and thank my funders. And I especially want to thank Christina Boucher and Travis Gagey. His name is cut off at the bottom there. Travis Gagey, my collaborators at Florida and Dalhousie University. Thank you.
You know, saying the R-index, which is only used within a species, there's a very hard to see if you go across the, presumably, you know, as you go up the tree, it still gives you some comfort, and that's not much, but
yeah,
do you have a sense of where that might start, right? And why should we not use it?
Yeah, it really is. It really does come down to a matter of compression ratio. The compression ratio does eventually suffer. And I would say the dividing line is probably somewhere between, we were talking about this earlier, somewhere between the kind of variation that exists between, say, humans or other eukaryotes versus the kind that can exist between bacteria. As I was saying to you earlier, if you sometimes, like if you were to look at the tree of life and extract a particular gene, like the 16S or 18S RNA gene from all over the tree of life, that does get a good, fairly good compression ratio with these approaches, but whole genomes just tend not to. There are other approaches, including one that a colleague at Dartmouth just preprinted, or at least I think the preprint's coming, that uses not quite the R-index idea, but a different set of index data structures that are geared toward the sort of midpoint between no compression and the wonderful compression we get with many human genomes. If you tweak the data structures a bit, you can sort of live in that space a little bit more comfortably. And so he considers that kind of like the future for if you wanted to use these tools for metagenomics. Questions on Zoom? Yeah, question.
I think not completely related, but are you working on like any hardware level optimization for this kind of stuff? Like if you like maybe beyond the point we will be able to optimize indexing.
Yeah. Absolutely. Yeah, I think if you look at people who've tried to hardware accelerate the BWT based stuff, they run into issues with unpredictable memory accesses, right? And basically you end up having, you know, your job becomes, you need to make sure the memory bus is fully used at all times by parallelizing many different queries so that they're all getting their queries into the bus, and you don't have to wait. You don't have to incur full latency of waiting for any one thread to go get something from memory. So I think this work, the movie work, it leads to, of all the BWT structures that have been proposed before, this is the one with the most radically predictable memory access pattern. Every iteration is going to be either one or maybe two cache misses, never more than that, because you can just analyze the things that are going on in the inner loop. There's only a couple opportunities for there to be a cache miss. So if you were going to implement it on hardware today, I think, first of all, you would start here. And then your next job would be how to spin up enough threads so that you can keep the memory bus completely full of queries. It's very unlike dynamic programming or other things that look like systolic arrays, and so you can put them down on the hardware really, really straightforwardly. This is nothing like that. It's really just about trying to optimize the use of memory, trying to not wait the latency for the misses in the main memory. But I don't know of any successful attempts to hardware accelerate BWT. I mean, I shouldn't say that. They are, I just don't know how much the hardware actually helps compared to if you had done it on a CPU. Yeah. Yeah, there's been a paper or two about ASIC designs for read alignment. Yeah.
Want to have the entire genome accessible to us, but at the end of the day, I guess dependent on what we are working on, we only look at a certain region across different populations. So have we also thought about, like, tackling this problem by just like storing a part of it? that would be required.
Storing a part of it. I wasn't sure what you meant by that.
Yes, like, just making the next to just get a part of it.
Oh, yeah. Yeah. Yeah, actually, in my earlier answer, I was talking about an experiment where we took just 16 sRNA and 18 sRNA genes from across. That's an example, right? Like you could imagine if you have in mind a relatively conserved gene, and your ultimate goal is to do something metagenomicsy, where you just sort of like to know what's the representation of the different of the species that have different alleles of this gene, then you could absolutely just extract that gene sequence. You would make a pan genome out of just that gene, you know, different alleles for that gene. In many ways, it should work super well for that application because presumably the compression ratio across alleles of a gene is usually better than the whole genomes that they occurred in, the compression ratio for the whole genomes they occurred in.
Thanks, Ben. Okay.